Viruses find clever ways in establishing infection, promoting replication, and enabling transmission. One such virus is Zika virus that caught the world's attention in 2016 when the World Health Organization declared an outbreak in South America as a public health emergency of international concern, as newborns of mothers infected during pregnancy had smaller than normal heads, because it is through developmental problems in the brain. Since then, cases of Zika virus infection have been detected in my home country, the Philippines, and in other parts of Asia, as well as warmer areas in North America. Although this virus causes disease in humans through bites of infected female mosquitoes, mosquitoes, on the other hand, seem to be unaffected by the virus because they have natural antiviral mechanisms to combat infection. However, these mechanisms are yet to be clearly understood. My research would like to understand how Zika virus evades mosquito antiviral responses and how it uses mosquito proteins during replication. By developing mosquito cells that express Zika virus proteins, I was able to learn how these proteins interact. These protein interactions form networks, quite similar to how online social media works. How? Think of it this way. A virus protein could be friends with a mosquito protein. They interact based on how their functions affect each other. An interesting aspect in this protein interaction is Zika virus using one of the most routine regulatory processes in the cell called ubiquitination. What is ubiquitination? Imagine you want to post a photo on your social media account. You take a selfie and use the hashtag #WokeUpLikeThis. We use hashtags to describe our photos and allow our photos to be curated online together with other photos having the same hashtag. Ubiquitination occurs in the same way. A small protein called ubiquitin is bound to another protein to tag it and let other proteins know what it is intended for. I have observed that ubiquitination is important during early stages of Zika infection by fooling cells into thinking that ubiquitinated virus proteins have an intended purpose in the cell. But actually, it just needs to be tagged so that it can start replicating its genome. Hashtag clever, hashtag expectation versus reality. This seems to be the case not only in mosquito cells, but also in human cells. This process is a potential target for Zika virus infection so that we can stop it and prevent transmission and infection. The weather in Scotland is not favorable for mosquitoes to thrive, but with climate change and increasing global temperature, we only expect for things to get worse in affected countries, and we expect for other countries that are not affected to have a potential threat. With this, we need to come up with novel solutions to outsmart that clever virus.